is sucrase.
Sucrose is a disaccharide made up of the monosaccharides glucose and fructose. Sucrase is an enzyme that catalyzes the hydrolysis of sucrose into glucose and fructose in water. Sucrase is involved in the digestive process and is often found in the small intestine. This enzyme has 4 subunits, and the active site is most often found in the center of the 4 subunits.
Sucrase catalyzes a catabolic reaction.
A catabolic reaction breaks macromolecules into its monomers.
Sucrose is the substrate that goes to the active site of sucrase. The active site is where the substrate binds to an enzyme so a chemical reaction may occur. Sucrose is a disaccharide that breaks into its monosaccharides, glucose, and fructose. Once again, let me emphasize this is most definitely a catabolic reaction, which is exergonic.
Although energy is taken in to hydrolyze sucrose, as a whole, energy is released. The energy released comes from the formation of other individual bonds between hydrogen and hydroxyl ions in water. This constitutes the fact that the reaction is exergonic.
After the substrate binds to the active site, induced fit occurs. This is when the enzyme changes its shape around the substrate to make the binding tighter until the reaction is complete. This increases the effectiveness of the reaction.
Enzymes help lower the activation energy, which is the energy it takes for a reaction to occur. This makes them catalysts, and they are used and reproduced in the reaction.
Sometimes inhibitors bind to the active site of the enzyme to block the substrates. These are known as competitive inhibitors, and they reduce the amount of products able to be produced by the enzyme. Non-competitive inhibitors bind to the other parts of the enzyme, which change the shape of the enzyme into a less effective form. If enzymes are overproducing a certain molecule, inhibitors help reduce the amount being made.
Thank you for watching this video about sucrase.
Enzyme, yo!